Identical twins unidentical after one spends a year in space. This is Mark and Scott Kelly, both NASA astronauts and identical twins. Well, at least they used to be. Following his year in space, scientists compared the DNA and RNA of Scott Kelly with his identical twin brother Mark via whole genome sequencing. They found that 7% of Kelly's DNA had changed into so-called space genes. As well as that, his immune system, bone formation, and eyesight all went under changes during his year in space. Telomeres, caps on chromosomes, typically shorten as a person ages. Kelly's lengthened but returned to normal 2 days after he returned to Earth in 2016. So, given a chance, would you spend any time in space? Genetic architecture.
Are humans next? China successfully cloned monkeys using transferred DNA, marking the first time such a feat has been achieved and possibly paving the way for human cloning. Scientists in Shanghai have cloned 2 genetically identical macaques using the same technique that produced Dolly the sheep. Somatic cell nuclear transfer involves taking the nucleus of a cell, which contains its genetic material, and injecting it into an egg that has had its own nucleus removed. The egg cell is then treated with enzymes to stimulate embryo development, just like a naturally fertilized egg. In all, the researchers created 109 embryos and implanted them into 21 surrogate monkeys, resulting in 6 pregnancies but only 2 live births. The macaques named Zhongzhong and Huahua aren't the first primates to be cloned, though. That distinction belongs to Tetra, a rhesus monkey created using a simpler embryo splitting method. The Chinese team believes the monkey clones could be useful in medical research, specifically in the study of genetic diseases like Parkinson's or autism.
Genes can help find time of death. Post-death gene activity may one day be a new area for forensic specialists to explore. A study from Spain has found that gene activity in dead bodies can point to the time of death. It found post-death activity in the brain and spleen to show little change. However, they also discovered post-mortem fluctuations across 600 muscle genes. The research was done via an algorithm that looked at samples from 399 people. The program attempted to tell the times of death for 129. In blood samples, it found that DNA production, immune response, and metabolism declined 6 hours after death. Most gene activity was found to occur between 7 and 14 hours after loss of life. If developed further, their approach could one day be used as a criminal investigation tool.
How to edit genetics without changing DNA. Scientific brilliance coming out of the MIT Harvard Broad Institute may one day better the lives of millions. DNA directs protein on how to build cells via RNA. A new study, published in the journal Science, details how DNA instructions can be chemically altered via edits to information in the RNA. The DNA remains unchanged, while proteins build cells based on the edited instructions. The method is not ready for human trials, but could one day help those suffering from degenerative diseases.
The future of data storage. Humanity is producing data at an exponential rate, prompting scientists to seek out storage systems that can keep up. Data is commonly archived using magnetic tape, which can hold as much as a terabyte per roll up to 30 years. But conventional storage capabilities may not be sufficient for long. Microsoft is now looking to DNA as a means to store digital data. DNA is more dense and can hold a quintillion bytes of information in 1 cubic millimeter. In its demonstration project in 2016, the company managed to encode 100 literary classics, or roughly 200 megabytes of data, into the 4 bases of DNA. But with the technology still in the early stages, converting digital bits into DNA code remains both slow and expensive. Microsoft aims to have a DNA storage system in place for at least 1 application in 3 years. The ambitious endgame, however, is for DNA to be the go-to for data archiving. Designer babies may soon be possible. A team of researchers at Oregon Health and Science University have successfully performed the first known attempt to create genetically modified human embryos. Gene editing using CRISPR technology is capable of finding a target DNA sequence and replacing it with a desired sequence. Once injected, the guide RNA in the CRISPR-Cas9 system seeks out the target DNA before the Cas9 enzyme binds and cuts it. The void in the DNA strand is then filled with the desired sequence. CRISPR technology theoretically makes it possible to modify the genomes of any living thing on Earth. Researchers in China have previously conducted trials using CRISPR technology in cancer patients, but the results revealed a major problem. The technique can cause unwanted mutations in other parts of a DNA sequence. However, the Portland team is confident that it is possible to avoid such errors.